to 1308 web series, Chapter 8, Cellular Reproduction, Cells from Cells. Now, Chapter 8, Chapter 9, Chapter 10, Chapter 11, Chapter 12, those chapters relate to the genetics, along with how cells reproduce. So Chapter 8 will be within that theme. We'll start with the first slide. Our first slide is this one. Okay. This has to do with the DNA. And if you recall, the DNA is made up of 4 nitrogenous bases. Those nitrogenous bases were the letters we saw before, A, T, G, and C. So that's adenine, thymine, guanine, cytosine. Those for their DNA. Recall from our previous chapter, RNA has adenine, uracil, guanine, and cytosine. So instead of thymine, RNA is going to have uracil. So it's A with U and G with C. DNA is always double-stranded, RNA is single-stranded. These are things we saw before. Now, why this chapter is so important is because one cell gives rise to another cell. So the information of one cell needs to be passed on to the next generation of cells, and that's what this chapter is about. Now, I know I've said this several times. I'll be saying it again in this chapter as well, so I might as well start now. Remember, your gene, your genetics, your gene is really a sequence of DNA that codes for a specific protein. Next slide. So here we have a term that we're going to look at, and this is a rare situation, so we're going to look at this one. It's called parthenogenesis, and this whole slide's about that. Parthenogenesis is when one sex, in this case the production of an offspring by a female without the involvement of a male counterpart. Here the daughter genes come solely from the mother. And only certain select animals have this ability. So you'll see like the lizards. There's some lizards that have this ability. The kimono dragon that you might see in this chapter, the kimono dragon or the Gila monster, they have this kind of ability. On this slide, it's a new one for me. I found out that female sharks also have this ability of parthenogenesis, where they can give rise to an offspring without the involvement of a male counterpart. So the daughter genes or the next generation genes come solely from the mother. It's called parthenogenesis. So here's an example of a certain kind of shark that has that ability. Okay, next slide. Okay, so one situation we saw a moment ago is parthenogenesis. Now, normally the reproduction occurs by cell division. So cell division, where one cell breaks off and now we have 2 new cells. Now, in order to have 2 new cells, we have to do what? We have to copy or duplicate the chromosomes that are present, so that way the 2 new cells will also have the same chromosomes. Okay, so we have to double the genetic information, double the chromosomes, and that's what this line says right here. Before a parent cell splits into 2, it duplicates its chromosomes. Now, if you remember correctly, I mentioned that chromosomes typically take on the shape of the X or Y shape. Now, chromosomes have our DNA, but remember what I said in a previous chapter. These chromosomes are only visible at the time of cell division. Before cell division takes place, the chromosomes take on a form I mentioned before called chromatin, which is loose spaghetti type of thing, which is not able to be seen without a microscope. Chromosomes you can, chromatin you cannot. So before the cell divides, it's called chromatin. After or during the cell division, actually, it's called chromosome. And we'll see that. Now, these are the reasons we have cell division. Cell division is to replace damaged or lost cells. So if you get a paper cut, well, on that paper cut, on that finger, for example, if that's where the paper cut is, that's where the cell division will take place, where you will have new cells grow to fill in the spot where the damage occurred. So that's one reason. So replacing damaged cells or lost cells, that's one reason for cell division. The next one we have is permits growth. So you don't have the same size hand that you had when you were a baby, right? So you're now growing, and that growth is coming from cell division. So you don't have the same size foot as you did when you were a baby. That's all because of cell division. So growth and repair. That's the kind of language you have to remember for a test. Growth and repair goes with cell division. So growth and repair. Then down here we have allows for reproduction. That's also a part of cell division. So we have growth and repair and reproduction. So growth and repair and reproduction. Next slide. And by the way, the one I'm on now, before I move to the next, this is an important slide. Cell division plays a role in one kind of role is growth and repair, and the other kind of role that cell division plays is reproduction. Okay, now I can move on. Now these are the forms of three functions of cell division. So in this picture here, we see a cell dividing because of replacement. Some damage has occurred, and now we're replacing the cells, so that's one reason. The other is growth. So that's this one right here, where the cells are growing. That's what tumor cells do, cancer cells. They grow like wildfire. So cell division, growth. Then down here, we have an amoeba, again, reproduction. Then we have another type of growth and reproduction, and that is a starfish here. So that's one kind, where if one leg of a starfish breaks off, it actually will grow an entire new body. And again, we are dealing with mitosis here. And then we have the last kind, where we are taking a piece of a plant. They call it clipping. So I'm taking a piece of a plant, and I've done this at home, where I've taken a cactus, a certain kind of cactus, and I saw that one end is growing too wild, so I actually cut it and put that little piece in a separate pot, and now I have two plants, one that's growing and one's already there. So again, that's a form of mitosis. Right? So three functions of cell division. So those are the same three that we saw a moment ago: growth, repair, and cell division. Okay, next slide. Now, this is where mitosis comes into two categories. We have asexual reproduction, as what we see on this slide right here. So asexual reproduction. And then down here we see the same comment again. What's mitosis for? It's for reproduction and growth and repair. So for multicellular animals like us, that'd be growth and repair. For other animals, it would serve as a purpose of asexual reproduction. Again, still a form of mitosis. Okay, next slide. Okay, now we have sexual reproduction. Now this one does require an egg and a sperm, and those are called gametes. So you can see right here on a test, gametes are egg and sperm. So gametes are egg and sperm. Now this is going to be different. For sexual reproduction, we have something called meiosis or meiosis. Now here the difference is for meiosis, meiosis is for reproduction only, as you can see on this slide, reproduction only. Now the thing about meiosis is that you're now giving each egg, each sperm, half the number of chromosomes. Now, of course, it's going to be randomized in terms of which half you always give, and that's why meiosis or meiosis gives you a genetic variation. That's important for a test. So meiosis or meiosis will give you genetic variation. Okay? It won't be a perfect even copy. So when that parent cell divides, each sperm, each egg is going to get a different genetic shuffle. Okay? So a different genetic shuffle. Okay? So it says here two kinds of cell division. Mitosis is for growth and repair. Meiosis is typically for reproduction. So this slide is important. Meiosis typically for reproduction. Mitosis is for growth and repair. Mitosis always gives rise to the same information in both cells, assuming, again, everything's normal. So mitosis will always give two new cells, and those two new cells are identical. That's mitosis. Those two new cells are identical. Same genetic information. That's not true for meiosis or meiosis. There, the two new cells are genetically different. There's a variation, there's a diversity. Okay, now we're on this slide. Now this one is dealing with eukaryotes and how mitosis and cell cycle works in eukaryotes. Okay. Now this line right here, where it says the number of chromosomes in a eukaryotic cells depends upon the species. Now, we're going to see a slide shortly about the number of chromosomes that different animals possess. One oddity is this: it's not the number of chromosomes that dictates the complexity of the animal. We have a Drosophila fruit fly that has more chromosomes than we do. As humans, we have 46 chromosomes. 46. Drosophila has something like 112 or 120 or something like that. Okay? Far more number of chromosomes, but notice a fruit fly is not as complex as a human. So the number of chromosomes does not dictate the complexity. Okay? Having said that, here's the slide that it goes with. So here's a koala that has 16 chromosomes, a giraffe that has 30, a mouse that has 40, a dog that has 78. So as you can see, the number of chromosomes does not dictate the complexity of the animal. Even this rat here down at the bottom has 102. So you can see it's not the number of chromosomes. It's what's in the chromosome that matters. Okay, next slide. Now this I've already said before. I'm saying again because you can see I'm repeating certain themes over. So chromosomes are typically made out of chromatin. Chromatin is simply unraveled chromosomes. And when do I see chromatin? When the cell is not dividing. So when the cell's not dividing, that's when we call it chromatin. When the cell is ready to divide, or during cell division, a chromatin turns into chromosome. And only during that cell division time period do you actually see chromosomes. Once the cell division is over, the chromosomes will unravel, like that loose spaghetti story I keep saying, back to chromatin. Okay, next slide. All right, here is a nice picture. This is from a plant, so it's going to be slightly different. But as you can see, the author has labeled it as chromosomes, because while the cell is dividing, you actually see the thick version of chromosomes. The neighboring cell is not dividing, and as you can see, it's loose spaghetti in there, and that would be chromatin. But only during the cell division does it become chromosome. Okay, I'll go back to this slide. You can see how chromatin is mentioned and is wrapped around tightly as a coil around the proteins. Okay, and we'll see that picture coming up in a moment. Now, this comment here is exactly what I'm getting at. Histones are the proteins that these chromatins are wrapped around. So chromatin, T-I-N, chromatin is wrapped around these histone proteins. And as you keep wrapping and wrapping and wrapping, the chromatin becomes a chromosome. So to show you that picture, it's coming up right here in this one. So here we have a picture that we previously saw in another chapter, and we see our loose DNA here. And again, a sequence of DNA is called a gene. And here we have a chromatin, because all of this is now being tightly wrapped and coiled and coiled and coiled around histone proteins. So chromatin is being wrapped around histone proteins. And as it keeps coiling and coiling and coiling, it turns into a chromosome. Chromosome. And you can see down here at the bottom, it says duplicated chromosome. And in the center, we have something called a centromere. So a centromere is in the middle holding the chromosome together. So a centromere is in the middle holding the chromosome together. It's almost like a tie wire or a zip tie holding the chromosome together. And that's a centromere. It's held together in the middle. Okay, next slide. Okay, don't care for that one. All right, this one I do care for. Now, what I said a moment ago is now on the slide. You can see it says centromere, where the sister chromatids are joined together. So there are copies of each other. So they're copies, and it says here, which are identical genes. So they're copies of each other, like a duplicate copy, and they're tied together and held together at that narrow waist called centromere. I'll go back to that picture. You can see it's held together, that center waist area called centromere. Okay, centromere. And these are duplicated chromosomes which have identical genes. So each strand has an identical set of information. Okay, next slide. All right, so again it says it's a full-fledged chromosome now, and each is now identical to the other. Now this word right here, chromatid, a chromatid is a single leg. So let me go back to our picture. So over here, see how they use it in parentheses? I know it's small print, but it says sister chromatids because each strand, there are actually two there that look like a Y, or an X actually, an X. And that strand and that strand are copies of each other, and they're called sister chromatids. And that's what makes up a chromosome. Sister chromatids, which is each leg, and each leg is now a sister chromatid of each other. So each leg. So if someone says on a test, what is a chromosome made out of? They're made out of sister chromatids, meaning each leg is a chromatid, and they're sister because they are copies of each other. And they're held together in the middle here at the waist, and that's called centromere. Okay, moving on. Okay, that's done. That's done. And here's a picture of it. So again, I've said it, and now you see another copy. So here is a chromosome that's been duplicated. Each leg is a chromatid. And when they split apart eventually, they're going to be called full-fledged chromosomes. And yes, they are copies of each other. So the one on the left will go to one cell eventually. One on the right will go to another cell. So when one cell turns into two cells because of cell division, each cell is going to get the exact same copy, same information. It's identical to each other. Again, we're talking about mitosis in this example. So the one on the left, it gets that information. The one on the right gets this information, and they're identical to each other. Okay, next slide. Now this is called the cell cycle, and there's a picture coming up. The cell cycle. Now the cell cycle is really important because every cell in your body has a cycle, a lifetime, a span. Right, from birth to death kind of thing. All right, and notice it says most of the cell's time is spent in interphase. We'll see that in a moment. Interphase is the biggest part, is the most time spent by the cell in interphase. That is when the cell is not dividing. So when the cell is not dividing, it's in what's called interphase. Now please don't make this mistake. Just because the cell is in interphase, it doesn't mean it's not doing anything. All it means is that it's not in the process of dividing. That's all that it means. There's a whole lot of stuff going on during interphase. You just don't see it because it doesn't look like anything's happening. But things are going on during interphase. So interphase is when the cell is not dividing. Okay, next comment. Now here's a picture version of the same thing. So the picture version of the same thing called cell cycle. If you see in this picture, most of the picture is defined as interphase, when the cell is not dividing. The only part when it's actually dividing is called this M, and M stands for mitosis. So it's only during this M when the cell is actually dividing. That's true for your skin cells, for example. That's definitely true for your brain cells. Okay? So yes, the skin cells tend to divide, but they don't spend the whole lifetime dividing. They only divide a certain amount of time. And your brain cells, they actually spend more time in interphase. They rarely divide. Now, during interphase, you see three parts. You see a G1, you see an S, and you see a G2. Now, in G1, which is called first gap, in G1, the cell is making materials for getting ready to divide. Okay, so it's getting things ready for that, making materials that are necessary. Then during S phase, that's where it's actually making a duplicate of the chromosome. So you got to make a copy of it. Okay, so you got to make a copy. Remember, you have 46 chromosomes. So 46 chromosomes in a typical human cell. Okay, 46 chromosomes. So we're going to make a copy of the 46 so that when we make new cells, the two new cells can have their own 46 when it comes to mitosis. Okay, so we're making copies of it. So one cell divides into two. You're going to see that each one now has 46 chromosomes when it comes to mitosis. So we're going to actually make a copy of it. And that's what you see in the S phase. DNA synthesis, chromosome duplication. That's S. So now you can see a copy made. Then in G2, that's where I am now. In G2, you're making materials for the actual cell division, which is mitosis. So you've got to make materials for it. Okay? And that's what you're doing during G2. Okay, next slide. This is what the author says. It says the same thing that I just said a moment ago, so I'll move on from there. Now we have the actual cell division part, which is mitosis. So the M goes to mitosis. Now here's the catch that a lot of people don't understand. Let me go back to our picture here. Notice that the author is calling this thing a cell cycle, but it only really labels only the interphase part. Mitosis is considered a separate entity, a separate thing. Okay, so most authors do that. Okay, so here we are with mitosis. So this is where the cell will physically separate. Okay, we're going to actually make that distinction here. This is going to be mitosis, where it says its contents divide evenly into two daughter nuclei. Now, the next word is very, very important for the purpose of the test, and that is cytokinesis. Now, notice the author says is divided into two. Well, that's because what's divided? The cytoplasm and all the organelles that go in the cell. So the mitochondria, the Golgi apparatus, the rough ER, the smooth ER, all those things that go as organelles in the cell, they have to be divided into two. So on a test, keep in mind that even though we are dividing the cell in mitosis, the actual physical separation is done during cytokinesis. I'll show you a picture of that in a moment. So the actual physical separation is done as cytokinesis. Notice it's not the same as mitosis. So mitosis is the genetic separation, where we've now separated out our 46 chromosomes between two cells. Okay, that's mitosis, when you genetically separate out the two sets of chromosomes, 46, 46. That's really mitosis. Cytokinesis is the physical separation of the two cells. So if you make two babies that are joined together, just as an example, two babies that are growing together but connected together, that part would be like mitosis. But when you physically separate them, that would be like cytokinesis. And we'll see a picture of that. Okay, that's a repeat. I don't care for that, just words. I do care for this picture. So this picture shows you the stages of mitosis. You will need to put this on a flashcard. So that's the best way to study this one, is putting it on a flashcard. So I would have this image on a flashcard. So if we look carefully, we see interphase as the first part. And then here you see that it's not called really a chromosome. It says uncondensed. Well, uncondensed or loose spaghetti is really chromatin. So it would be really good if the author had actually wrote chromatin here, because it looks like loose spaghetti. So when they say uncondensed chromosome is chromatin. It's that loose spaghetti. And then we have our 4 blue colored steps here called prophase, metaphase, anaphase, telophase. Why are these headings in blue color, whereas interphase was in aqua green? Because cell division, mitosis, is not the same thing as interphase, right? We saw that. So this is true mitosis: prophase, metaphase, anaphase, telophase. And in prophase, the cell is starting to divide. And now the author calls it what? Condensed chromosomes, or true chromosomes. And I have already said many times, you see chromosomes when the cell is dividing. And that's when we have prophase, metaphase, anaphase, telophase, during the actual cell division. That's why it's called chromosome. Now in prophase, notice we're doing something. We're creating something over here called a mitotic spindle. And what's doing here is creating like a highway or a track that's going to open up or separate out the pieces that go with our chromosome. So over here, we've made our chromosome, and you can see it says chromosome now. And then we're now going to line them up, and it truly lines up during metaphase. So on an exam, it says, when the chromosomes line up at the equator. Well, that's what they mean here. They mean metaphase, where all the chromosomes line up on this equator. And that looks like to somebody an equator. And then what happens? You separate them out. And now that's anaphase, when you're actually separating out the chromosomes. So when you're separating out the chromosomes, that's anaphase. And then we end it with telophase, where now we have copies on each side. One on the left for one cell, one on the right for another cell, and notice the nucleus is now reforming. See how over here on the left under prophase, the nucleus went bye-bye. See, there's no more nucleus. The nucleus went bye-bye. Then in telophase, the nucleus came back. So the nuclear envelope came back. Now what's missing on the far right of this slide here? That's not even there. What's missing is cytokinesis. Now that's not a mistake by the author. Cytokinesis is not truly mitosis, and that's what I was getting at on the other slide. Cytokinesis is what comes after mitosis here on the edge somewhere of our slide. And cytokinesis is the physical separation. So if I actually cut this, that would be part of cytokinesis. But remember, it does not take place in the actual cell division by definition. So if I were to go down below on this slide here where all this structure stuff is for people, well, it would say cytokinesis is a physical separation. It's a step that comes after mitosis. So if I were to go back to our slide that looks like this, somewhere in this mitosis here, even though it's not shown as a separate thing, there is a cytokinesis built in. Technically not part of it, but it's built into mitosis. So that's why you don't see it here, but it's actually part of mitosis. But why is it not officially part of mitosis? Because mitosis is the separation of the genetic material, and cytokinesis is the physical separation where we actually take these two cells and split them apart. Okay? Now what I would do for this is you can either use this slide down here, or you can use the PowerPoint that I'll show you in a moment. Those have the details for each of these steps. And so I would put them on a flashcard. The image here is nice. So if you just want to make a copy of this page and then put it on a sheet of paper and then or on the flashcard, that would be nice too. That would be helpful. Okay. So now here you can see that the author has said that mitosis is one thing and cytokinesis is something different. It comes after telophase. See how it says usually begins during telophase, but it really finishes after telophase. And like I said when I was on this slide, cytokinesis would be somewhere off of this slide to the right. Okay, next slide. So it's the physical separation when it comes to cytokinesis. Okay, and here's an example here as well. Okay, cytokinesis, physical separation. All right, here's the animal version of cytokinesis where it splits and then you get two new cells. So remember, this is not part of mitosis. It's a separate act that happens afterwards. Cytokinesis, where we physically separate out the two cells, cutting them separately. Okay. Now here's a plant version. So this was the animal version that you currently see on the screen. Plants are different. In plants, they have something called a cell plate that forms. So in a plant, you have a cell plate that forms. Let me show you a picture of that. So here's the author's version for plant cell cytokinesis. Here, I see a physical plate forming. Remember, only plants have cell walls. Remember that chapter I told you to make flashcards for the parts of a cell for both animal and plant? Well, you know plants have a cell wall, but they actually have something called a cell plate that forms between them. And then you get the two new cells from that. Okay, next slide. Okay, now, this is where I mentioned going round and round and round the cell cycle. So the title I'm on now, it says cancer cells. Let me go back to our other picture. And the picture I'm going back to is this one. This is how normal cells function. But what if the cancer cell shows up on this slide? The way cancer cells would look is that this mitosis part wouldn't be short. It would be a longer duration. It'd be more often. And this cycle would go around faster. Normally, skin cells divide every 30 minutes or something like that. Each skin cell divides every 30 minutes. For example, I have a corneal abrasion, and it's really epithelial cells on the surface of my cornea of my eye. And since those are epithelial cells, just like the skin cells, they're going to divide rapidly. So it doesn't take too long for that to heal. Okay? All right? So that's why cancer cells operate that way. So let me go back to our slide. And now we see that the problem with cancer cells is they don't know when to stop that cycle, that picture that goes round and round and round. And cancer cells just grow uncontrollably. That's why it says dividing out of control. They don't know when to stop. All the signals that say, hey, this is only so many times you get to go around that circle. Well, with cancer cells, they're not told when to stop. So they keep going around that circle, around and around, not every 30 minutes or much faster than that, because they don't know when to stop or when to go during that nice little circle. Okay, and that's why cancer is a disease of the cell cycle. I love that line right there. Cancer is a disease of the cell cycle. It really is. Cancer cells just keep going round and round in that circle too fast. They don't know how to pace themselves because the signals that say when to stop, when to go are broken. Okay? And that's why these changes cause the cell to grow abnormally. And what happened is somehow the signals that are telling them how often you should go around that circle, those signals are damaged. And it says here, it begins when single cells undergo mutations. And let me now explain that word mutation because it's going to come up from now on. A mutation is a change in the gene. Now, you know how many times I've given you the definition of a gene, so I'll do it one more time and then I'll explain what mutation is. A gene is a sequence of DNA that codes for a specific protein. And then when you have changes in that gene, which are called mutations. Now keep in mind, a mutation is typically a permanent change, because we have a lot of mechanisms that go around and do spell checking, for example, to make sure all the letters of A, T, G, C on that gene, on that DNA are correct. And if you remember from a previous chapter, I showed you a picture of a normal red blood cell hemoglobin and the sickle cell shape hemoglobin. And the only difference at that time in that chapter that I pointed out to y'all was one letter difference. Well, that is true here too. You can have one letter difference, but it's a permanent change. So a mutation is a permanent change. We have changes going on all the time, but we do have spell checkers out there that are designed to double check to make sure all the genes have the correct letter. So if an A is supposed to be an A, but instead it turns out to be a T, there are spell checkers that go there and fix the T back to an A again, for example. Okay? So that's why we don't come down with cancers all of a sudden all the time. There are certain steps that actually are required for a cancer to happen. It doesn't happen all the time in every single situation. There are reasons for it, because mutations have to be in a certain way. And mutations have to be permanent. So it has to be a permanent change that causes the cancer to take place. Okay, next slide. Now here's our what is cancer. First of all, cancers form tumors. So that's an abnormal growth of these cells. That's what it is, abnormal growth. Now you can have a benign tumor, and a benign tumor doesn't spread. It just stays in one place. It's not going to change unless something weird happens, but it is a benign tumor. It stays in one place. Okay? And that's easy to fix because all you do is just cut it out. Okay? So a benign tumor stays in place. It's an abnormal group of cells. Okay? Not good, but at least it stays in place and it doesn't spread. Okay? Then we have malignant tumors. Now, by definition, malignant tumor spreads to other parts of the body. So that's bad. So malignant tumors will spread to the other parts of the body. Now here's the bad part of malignant tumors. When it goes to another part of the body, that new area changes in cells and everything. That new area can actually be worse than the original cancer. So when someone has colon cancer and it spreads to the lungs, now the lung cancer can actually be worse than the colon cancer that they had initially. So that's why malignant tumors are bad, because they can spread. So if you have a colon cancer and that cancer spreads by the blood to the brain, and now you have a brain cancer is spread like wildfire in the brain. So it's very, very difficult to treat a brain cancer. Right? So malignant tumors spread from here to there. Now when they do spread, okay, that is really a true cancer, because by definition, a true cancer is a malignant cancer, the one that spreads. The spreading of these cells is called metastasis, and that word is there. You can put it on a flashcard if you wish. That's metastasis, where it spreads. Now, there are two ways it spreads, and I'm going to give that to you for the purpose of the test. They do ask this on the test. Two ways it spreads. Cancer spread by blood, and they spread by lymph. So that's what's shown in this picture here. They spread by blood, and they spread by lymph. So if someone has a breast cancer, they have to remove the lymph nodes that are in the armpits, because if the cancer does get there and spreads to the other parts of the body, that's a problem. So cancers that spread, they spread by blood or they spread by lymph. Okay? So that you need to know for the purpose of the test. And it says it nicely down here. Cancer cells spread throughout the body by lymph or blood vessels to other parts of the body. And then now you have tumors elsewhere. And when they do take on a new location, they take on a different kind of cancer. Okay, so blood and lymph is important. Okay, then we have treatment. Two kinds of treatment. We have radiation treatment so far, radiation treatment, and then they have chemotherapy. Radiation's from the outside, where they zap you with radiation, hopefully to kill the tumor cells. And the other is chemotherapy, where they give you drugs into your body, and then they're trying to fight it that way. So they're fighting it from inside instead of the outside. So whenever you get your x-rays taken at the doctor's office or the dentist office, that's why they make sure you don't get too much radiation. Okay, let me give you an example before I go on. When I was flying on a flight once, I unfortunately had the seat in the very, very back on one of my flights. And it was a pilot that was sitting next to me. He was, of course, just traveling from place to place. And he ended up having cancer on his nose. Pilots are very prone to cancers on their faces because of the location of the plane and because you don't have all that atmosphere because you're flying at 35, 40,000 feet and you're much closer to the sun, and you're also away from the protection of the air layers that normally would protect you when you're on the earth or when you're on the ground. So that's why radiation for pilots, for people who fly a lot, they get a lot of cancers on their skin. So he had a skin cancer on his nose. Okay, I don't really care for this one. Okay, these are the things that you can do to prevent and survive. One, smoking. You want to cut down on your smoking, so don't smoke. Second, exercise. You want to exercise. You do want to avoid overexposure to the sun. Our actor that most of you might know is Hugh Jackman, who's from Australia. He's the one that plays the Wolverine guy in X-Men or something like that. Well, he had skin cancer on his nose because a lot of Australians like to go sunbathing, and so it's a really dangerous thing to be out there too far often in the sun. Okay, so avoiding overexposure to the sun. Eating a high-fiber, low-fat diet. So if you're always a McDonald's fan all the time, eating a lot of fatty foods, that's bad too. You want to eat a high-fiber diet to help move things through your colon. Then performing self-exams to just make sure you don't feel anything. And then visiting your doctor regularly to check whether you have any issues. When you get to an age of 50 plus, for example, I think now it's gone down to 40 plus, but if you're 50 plus, they ask you to do that little Cologuard test where you just check in your stool, you give a sample of your stool through the Cologuard program, and they mail you a box, and then you send off the box with your stool sample, and they see whether you have any colon cancer. And yes, there are some false positives, false negatives, and things like that, but it actually is pretty good at detecting early stages of colon cancer. And that's one of the easiest cancers to take care of. So you want to make sure you're checking yourself. Okay, then we now move over and switch over to a different form of reproduction, which is meiosis or meiosis. And remember, that one's going to be the sexual reproduction. So meiosis or meiosis. And remember I told you, this is where the parents give, through the egg and through the sperm, they give their genetics. Now when the egg and sperm fertilizes, you again get your 46 chromosomes. So each egg has 23 and each sperm has 23 chromosomes. And when the 23 fertilize, you get your 64 again. Or sorry, 46 again. So that's how sexual reproduction works. Now here's the catch though. Because each parent is giving a certain kind of egg and a certain kind of sperm, each egg is different and each sperm is different because of meiosis. So when you start off with 46 chromosomes and each egg has 23 and each sperm has 23 chromosomes, those 23, each of them for each egg, each sperm, are going to be different. So yes, your parents are actually giving a shuffle to each egg, to each sperm, and therefore which sperm gets with which egg plays a big role. And that's what the next picture is about. So in this picture here, you see a blended family, and you see how the kids look like a mixed culture, all because of just that. The parents and the kids are going to be genetically blended. And that blend comes from the fact that one of his sperm, one of her eggs, and it could be a different egg, different sperm, doesn't matter. Eventually, they're going to have different kids. So each time they have a child, it's going to be a blend of whatever the parents are giving. And again, the blend is going to be shuffled every single time. So if I gave you a deck of cards, then I shuffled them, then I gave them back to you, and I shuffled them again. Every time I give it to you, it's going to be a different sequence of shuffle. That's exactly what this is like. So each egg is different, each sperm is different, and therefore each zygote, which is a fertilized egg, that's going to be different too. And that's why I said earlier that gives rise to genetic diversity. So meiosis or meiosis will give rise to genetic diversity. So when we have a blended culture now, because at many, many places we have blended cultures getting married, well, if that's the case, their kids are going to be different too. Okay, the next line here tells you that every human cell, not counting the sex cells, so not counting sperm, not counting egg, all the other cells have 46 chromosomes. And when you have the egg and the sperm, they're going to have 23 chromosomes. Again, they're going to be shuffled, so you don't know which egg and which sperm. Each one is going to be uniquely different. Okay, next slide. Now, I'm about to show you a picture. This picture is called a karyotype. You can see the word on the current slide called a karyotype. I used to do these when I first started in lab long, long ago to produce a karyotype. And what the person does is it takes a cell that's actually dividing. Remember, during cell division, we have one stage called metaphase in mitosis. When that happens, you can see all of the 46 chromosomes lining up, and then you take a picture of it, and then you cut them out. And when you do that, you end up with this. So right here is a karyotype. Now, how do I know that this is a male karyotype? Because I look at the X and the Y for the 23rd set. So you can see 22 pairs are perfectly paired off. But then the 23rd pair is an X and a Y. That tells me it's a male. So X and a Y for the 23rd set tells me it's a male. Had it been a female, it would have been an X and an X. So XX is for the female, XY is for the male, for the 23rd set. Now in each one of these, let's look at set number 1. That's actually an X right here. It looks like it's crushed, but it's X. This one's also an X chromosome shape as well. Again, remember it said centromere? That's at the waist. So we have one duplicated chromosome, another duplicated chromosome, and these are sister chromatids, just like I mentioned before. Now here's the catch though. For every set, see how there's 23 sets. For every one of these sets, let's take the first set, the one that they've highlighted. The one on the left came from mommy. So the one on the left came from mommy. The one on the right came from daddy. How do I know? Because I'll show you in a moment how the XY works out. So this one right here came from mommy. This one came from daddy. For set number 2, the one on the left came from mommy. The one on the right came from daddy. Mommy, daddy, mommy, daddy, mommy, daddy, and so on. For every one of these sets, the first one came from mommy. The second one came from daddy. Now, how do I know? Because that Y is determined by the daddy. So if you end up getting a boy as a child, when you have a son and you have a boy, that boy is because of the father. So the father determines the sex of the child. Mother provides the X, and the father can either provide an X or provide a Y. So only the father can provide an X or a Y. If the mother provides an X and the father provides an X, then you're going to get a baby girl. If the mother gives an X, because that's all she can give, but the dad gives a Y, then that's going to be a son. It's going to be a boy. So in this case right here, this person, the X came from mommy and the Y came from daddy for this particular person. Okay. All right, so that's how that works. Now, why is karyotype helpful? Well, right here in set number 21, if he had an extra one right there for set number 21, that would be called trisomy 21, commonly known as Down syndrome. So commonly known as Down syndrome. And that's what tends to happen when chromosomes don't break off correctly. So you end up getting an extra chromosome when you shouldn't. And that's why having extra chromosomes can be a big deal. We're supposed to have 46. We're meant to have 46. If you have more or less, then that can be a big dramatic difference, and that can be a problem. Okay? So trisomy 21 will result in Down syndrome, and that would be a bad thing. So yes, not only can you tell the gender, which I did a moment ago with y'all, so you can tell the gender of the person, but you can also tell certain genetic information, because if you have more of the set or less of the set, then you can tell that there's a problem. The other thing is that each one of these, like see how they're color-coded? Well, they're color-coded based on the trait and the banding pattern. So see how the banding patterns match? The green, the yellow, the pink, the yellow, the green. And notice for every one of these, they're matching in color coding because they have common traits. So if the red at the bottom here for the mommy's one, let's say that's eye color. Well, then the eye color is also true on the daddy's one over here at the bottom. And that's how you decide who is giving a greater effect of the trait. Did that trait come more from daddy or more from mommy? So if you have a son that's lefty, well then if he's a lefty and it has a normal tendency to be a lefty, there's a reason for it. It must have come from one of the parents. Okay, so that's how that works. Next slide. Okay, these are called homologous chromosomes. So what I just showed you in the picture are called homologous chromosomes. Look what it says. They resemble each other in length. They resemble each other in banding pattern. So resemble in length. They resemble in banding pattern. And they carry similar traits. Let me go back one more time. Remember how I said the bottom? If that's eye color there, then it's going to be eye color there. So eye color from mommy, eye color from daddy. And so these are called homologous chromosomes. See how the length is the same between these two? See how the length is the same between this set, or even down here for number 20? See how the length is the same? The banding pattern is the same, and they have identical traits. And that's what's called a homologous chromosome. Okay, now I've already shared this with you too. I told you that we have 22 pairs, and those are called autosomes. So first 22 pairs are called autosomes. The 23rd pair is the sex chromosomes. That's the 23rd pair. That could be an X and a Y, or be an X and an X. So you can see here, females are 2 X's, males are an X and a Y, which I mentioned a moment ago. If I go back to our picture one last time, see the first 22 are autosomes, and the 23rd pair is the sex chromosome, X and Y in this example. Okay, next slide. Alrighty, I don't care for this because it's just too many words, but I do care for the picture that goes with it. So here we have an individual. So that's one person that they're talking about. 46 chromosomes. Then each egg, each sperm will have 23 chromosomes. And remember what I said, because of meiosis, which is what we're on now, because of meiosis or meiosis, each egg will have 23, each sperm will have 23, but the 23s are not the same, meaning each egg has a different set of information among the 23 chromosomes, and each sperm has a different set of information among its 23 chromosomes. And when a sperm and an egg get together during fertilization, they make a zygote. And a zygote is a cell with 46 chromosomes. That's what we have. So you can see over here, we are 46 chromosomes, each person. Same way here, 46. And the only time we have growth and development is done by mitosis. If you have a paper cut, that repair is done by mitosis. When you have little feet and now you have a big feet because you're an adult, that's also development. Okay, next slide. You do need to know these terms before I move forward. Haploid is half the number and diploid is the full number. So 2N is our diploid for us as 46 as humans, and haploid would be the 23. So be aware of that. Diploid is the full set, haploid like half. Haploid is the half of it. Okay, we've already seen the word zygotes. If I go back to our picture again, a zygote is a fertilized egg. So a zygote is a fertilized egg. Okay, next set. Okay, now we have this slide. How meiosis cuts it in half. So here we have a different story. And yes, meiosis or meiosis is like mitosis, but it has two parts. As you can see, there's a meiosis 1, and then there's a meiosis 2. So meiosis 1, meiosis 2. In meiosis 1, we're doing what? We are lining them up. So these are our homologous chromosomes, lining them up, and then we're opening them up. So meiosis 2 here, or meiosis 2, that's like mitosis. Okay. So meiosis 2, or meiosis 2, is more like mitosis. That's what I tell a lot of people. But meiosis 1 is unique. Here, it depends on how you break it apart. Just in this example, it just happens to be red and blue. So all the reds went this way and all the blues went this way. That won't be usually the case. This is a very simplistic view here, where the blue just went there. All the blues went that way, all the reds went that way, and then you ended up getting a red, red, blue, blue. That's not going to happen typically. Instead, it's going to be a variation. So let me give you an example of that. Okay. All right. Now, here's where the variation occurs. In meiosis 1, we have something called crossing over. So in meiosis 1, we have something called crossing over. How do I know it's meiosis 1? Because it says right here, first phase of meiosis. So in meiosis 1, we have crossing over. Now here's the magic about crossing over. In crossing over, if you look carefully, and I know it's too small to see this, but here we don't have this story. See how it says all the reds went there and all the blues went there? That's not what this picture is showing. In this picture, there's a blue and a red, and you can see it a little bit better in this picture over here, how there's a red and then there's a red and a blue, a red and a blue, a red and a blue. So there is a piece of the chromosomes that have exchanged places. And if you look carefully in this picture, you see the same thing. There's a blue with a piece of red, and there's a red with a piece of blue. This is what allows for genetic diversity. So if I went back to this other picture, and I did a little bit of mixing right here, where some of the red went over there, meaning some of the blue, let me back up. Some of the blue went over to the red, and some of the red went over here, I would have what? A red and a blue combo, a red and a blue combo, which would give me some combo of red and blue, blue and red, blue and red. That's where you get the genetic diversity. So meiosis or meiosis gives you more genetic diversity. If you look carefully again, there are two stages. There's our usual interphase where we have our chromatin, loose genetic material. Then we have our first stage of meiosis where crossing over and all that happens. And then we have our usual meiosis 2, which is basically like, in essence, like mitosis. So meiosis 2 is sort of like meiosis, where we're going to just have 4 split up eventually. And yes, there is cytokinesis here. Notice the author does say telophase followed by cytokinesis, telophase followed by cytokinesis. Now, when I said earlier about this red mixing with blue and blue mixing with red, well, guess what? How much of that crossing over happens is a random act. You cannot predict how much of the crossing over happens. So that's why when you have kids, you don't know how much of you, meaning mommy, ends up in your kid. You don't know how much daddy ends up in the kid. So you can have a child who is more like mommy, who's lefty and all those kinds of things, but then have different characteristics from the father. So the child could be either 50-50 parent, 20-30, 65-64, different variations of parents contributing to the child. That's because of crossing over. All right, going over here, notice it says the same thing. So you have the same information here that you can read. And now here's another picture of how mitosis compares. I already told you mitosis is for growth and repair, and it always gives you the exact same thing between the two cells. So notice the exact same thing between the two cells. That's mitosis. Meiosis or meiosis, after meiosis 1, we get some crossing over there. And now you can see down here that there is a red and a red and a blue and a red, and a little bit more of a blue and a red, any possible combination thereof. So meiosis is for genetic diversity, more variation. Okay, this says the same thing. Okay. And remember, mitosis always gives you the same number, whereas meiosis is always half the number. So over here, you can see it says 2N, 2N. So these are the same as the parent. So they're exactly the same as the parent. Exactly. That's how cells repair and grow in our body. Whereas for meiosis, for sexual reproduction, you see that down here they're half the number of chromosomes. There are 23 for humans. Okay, that's what that slide's saying. I don't care. I don't care. I do, but we're going to do it a different way. So let me go to the next slide first. Okay, now on this slide, and this is where it's going to get really important because this carries over to a future chapter. If you look carefully, this is part of meiosis 1 and that crossing over business during meiosis 1. See how all the blues are lined up on the left, all the reds are lined up on the right? Well, over here, the other possibility is a blue on the left, red on the right, then a red on the left, blue on the right. See, they're not lined up the same way. Not all the blues are on the left and all the reds on the right. There's a mix and match. Well, as a result, look what happens. You get more variation. And that's what this last term, let me go back to the slide, says independent assortment. You want to jot that down because that's going to be a big theme in a future chapter. Independent assortment, which says that the chromosomes line up independently of each other. So let me go back to our picture. So the rule or the law is called law of independent assortment. Okay? And notice the title, genetic variation. So one of the ways, and I got to say this over and over, one of the ways of genetic variation is independent assortment. And that goes to our picture here where the chromosomes can line up independently of each other. Where the blue and the red don't have to line up necessarily like that. They could end up being a blue and a red and a blue. And when these independent assortments happen, the way they line up alternative ways, you get more possible combinations. You don't just get blues and the red separately. You get a red and a blue, a red and a blue, a blue and a red, a blue and a red. So you get more possible combinations. And that is why meiosis gives you genetic diversity, because of independent assortment. Okay, next slide. Now you can see where the slide says independent assortment. Each egg, each sperm has 23 chromosomes, but if you look at math, that's 8 million possible combinations. And when you have the female and the male, because remember, male has a possibility and the female has a possibility. That gives you 64 trillion possible combinations. So when it comes to this picture, there are 64, let me go back to my number again, 64 trillion possible combinations when you do it this way. So the law of independent assortment gives you more variation. It's not just cookie cutter where blue's on one side, red's on one side, and you always end up getting the blues together and the reds together. Instead, it's going to be more like possibility number 2, where the reds and the blues, these homologous chromosomes, can line up in so many different ways, giving you more possibilities. And again, that gives you 64 trillion possible combinations. And that's why meiosis gives you so much diversity. That's why it's so difficult to predict exactly what traits you passed on versus the other person. Okay, here's fertilization happening where the sperm and the egg get together. Okay, and I've already told you this. I told you crossing over occurs during prophase 1 of meiosis. So we've already seen that in the picture form. Right, so during prophase 1 of meiosis. And we've also said that this combination of different crossing over possibilities, more crossing over, less crossing over, that gives you more recombinations or more genetic possibilities. So crossing over is one thing, and the law of independent assortment where the chromosomes line up. So let me recap and then I'll move on. So there's 2 things here. One is the way the chromosomes line up. That's called law of independent assortment. That's one way of getting variations or genetic diversity. And the other is crossing over. Remember I told you crossing over occurs during prophase 1, but we don't know, there's no way to predict how much crosses over. It's a luck of the draw. Okay, it's a luck of the draw as to how much is going to cross over. Well, that's exactly what this is like. And that adds further to the genetic combinations that are possible. Okay? So it adds more to the complexity. Next slide. So here's an example of crossing over where we have now a blue with the pieces of red. We have red with the pieces of blue. How much of the red turns into blue and how much of the blue turns into the red, that's because of crossing over, and that's luck of the draw. We don't know how much of the red is going to pick up blue and how much of the blue is going to pick up the red. Okay, we don't know. We also don't know this combination up here, which I mentioned, the law of independent assortment. It doesn't have to be blue and red together. It could be red and red or blue and blue. So there's many, many possibilities here. Okay, next slide. I don't care. Don't care. Okay, now comes this one. All right, so again, here we see these rotifers. These are pretty cool creatures. These rotifers move around. Well, they too have what? A sexual reproducing way of doing things. Okay, I really don't care for that slide. Yeah, okay. All right, still don't care about the rotifers. All right, now comes to something I have already talked about, but that's a boo-boo. Remember how I said that we can have trisomy 21, where we have an extra chromosome for the 21st set? And I said if you have an extra chromosome, that can be a problem, that can cause big issues. Well, that's where this comes into play. Non-disjunction, that's what it's actually called. So when chromosomes fail to separate properly, you get what's called non-disjunction. So when chromosomes fail to separate properly, you get non-disjunction. Notice it says how accidents during meiosis can alter the chromosome number. And that's where I said that if you have an extra pair or extra set of 21, the 21st pair, trisomy 21 will result in Down syndrome. And the problem is called non-disjunction. So if I go to the next slide, you can see it. So instead of having only 2, so instead of having, let me go back real quick. So instead of having just two, so I can see how there's only two, only two, only two. Well, for trisomy 21, if you have an extra pair, that's a problem. And that's caused by non-disjunction. And in this picture here, you can see that you have extra chromosomes, and here you have less. So that's a boo-boo. That's a problem. So here we see exactly two, but here we see an extra, and that's bad. So if I go to my slide on the next one, you can see how you have extra chromosomes. Extra chromosomes. And that's caused by non-disjunction, and that's bad. Now, I told you about Down syndrome, so there is a specific slide for that. So in a condition called trisomy 21, the 21st pair has an extra chromosome. So the 21st pair, or the set number 21, has an extra chromosome, and that causes Down syndrome, which is usually a mental retardation. They have a certain look. So let me go ahead and go to that next slide so you can have that look there. And I know this is small, but it says it right here at the bottom, showing trisomy 21, where there's an extra chromosome at the bottom. So there's an extra chromosome for the 21st set, and that's a problem. So it does cause mental retardation, scoliosis of the spine. They have a look like this, where they have the moon face sometimes, as they call it. Okay, so trisomy 21 Down syndrome. And it says here they have a shorter lifespan, usually mental retardation, scoliosis, have trouble walking, things like that. So it's a problem. Okay, next slide. I don't care for this one. I do care for this chart right here. So this chart does get asked. And this has to do with extra chromosomes. So if you have an extra one for the sex chromosome, for the 23rd pair, so if the 23rd pair, you have an extra one. Now the two that I care for are the top and the bottom. So the top one is when you have an extra X, so XXY, and that's a Klinefelter. Now the problem is, typically they are a male. So they look like a male, but notice what it says. They have they're sterile, so there's no fertility there. They're sterile. They have underdeveloped testes. They have secondary female characteristics. So Klinefelter's is a male with more female characteristics. Okay, so Klinefelter's. Now I would do a suggestion here. I would look Klinefelter's up on Google and see what the images provide you with. And that way you can see what a Klinefelter syndrome person looks like. So I would Google Klinefelter's and go under Images, under Google Images tab, and then you'll see what they look like. They are male, but they have more female characteristics. So that's Klinefelter's. Then on the bottom one, I have XO. XO is a female, but it has more male characteristics. So sterile again. Okay, so they're going to be sterile again. So they are a female, but they have more manly characteristics. Okay, so they're sterile and immature sex organs. So do look up Klinefelter's male and what they look like, and look up Turner syndrome female and see what they look like to get a better picture instead of just having a chart. Okay, next slide. So we are summing up with this slide here. We see asexual reproduction, where it doesn't need a partner. And that'd be like a starfish, for example. If an arm or a leg, however you want to call it, gets cut off on a starfish, it can grow a new one. And if the leg is there by itself, it can grow a whole new body. So that would be asexual. Then for sexual, we have advantages here because you can produce an offspring of varied genetic makeup, and it also reduces the incidences of harmful genes more rapidly because it's a little bit tougher to get that. So if you screw up on asexual, then the next partner will mess up, and the next partner will mess up because the problem will just perpetuate. It doesn't tend to happen with sexual reproduction. Okay? And again, sexual reproduction allows for genetic variation, whereas asexual will not, and nor does it help with anything else. Okay? So this slide is something just to be aware of. Next slide. Here's a plant. And this is an asexual way of growing another plant. This is like my clippings example that I gave earlier. All right, so for this chapter, make sure you're brushed up on this one. So if you're comfortable with 8, it'll help you with the remainder of the 3rd unit, which goes after chapter 10. So it'll help you with that. Okay. All right, so that takes care of chapter 8. You're ready to move on to chapter 9.